this is MCB2 tutoring. This video is about lipoproteins. Actually, I'm going to make this into three different videos, and we're going to talk about lipoproteins. So the way I'm going to organize these is this way. So the previous video, we talked about lipid digestion, and when lipid gets digested, it's made into chylomicrons in the GI. So the first thing we're going to talk about, so overview here. So first thing, we're going to talk about these chylomicrons. So this came from GI, from GI. And then their job is to drop off triacylglycerides. And then eventually we'll talk about they end up in the liver. Okay, so this first video we're going to talk about chylomicrons. Second one, we're going to go through the whole pathways with the VLDLs and then IDLs, and then eventually LDLs. Okay, third video, we're going to talk about the role of HDLs. So basically these are the three types of lipoprotein. So and they have a very distinct function. So it makes sense to kind of separate them and look at them in terms of chylomicrons, VLDL, and HDL. Okay, so the first one, chylomicron. The chylomicrons came from the GI. So recall, GI, you're absorbing a lot of nutrients, so you end up with lots of triglycerides, lots of maybe phospholipids, and then cholesterol ester, and then they're all packaged into along with ApoB48, and then they all become this chylomicron. And ApoB48 came from this gene that's B apoprotein gene. And this same gene is capable of making two products. The other one is a B100, which we'll talk about later in terms of the VLDL. So for now, we have a chylomicron. So this is a chylomicron, and it has this one copy of ApoB48, which is made in the GI. Okay, so this whole thing came from GI. And the main constituent of this chylomicron is the triglycerides, or the triacylglycerides. So I'm going to write this really big. So that's like 90% of the chylomicron. So it's this thing here is the main component of the chylomicron. When it's first made, it's considered a nascent chylomicron. And sorry, this one also has some cholesterol esters. Okay, so when it's first made, it's considered nascent chylomicron. And in order to become a mature chylomicron, so next step it would become a mature chylomicron. And for this to be a mature chylomicron, it needs to have a few more apoproteins. So it's going to get these from an HDL. So HDL is going to give it a few things. So HDL is going to give it ApoC2 and an ApoE. And we talked about a little bit here, the job of the chylomicrons is to drop off triglycerides, right? So it basically has to give off triglycerides. And then as it gives off triglycerides to the peripheral tissue, it's going to become a chylomicron remnant. So chylomicron remnant is basically going to have very little triglycerides, and it's still going to have the cholesterol and cholesterol esters. Okay. And then that basically is the lifespan of this chylomicron. And then at this point, it's going to get uptaken by the liver. So recall the chylomicron has an ApoE on it. And this ApoE is what's going to help bind to this receptor on the liver. So this is liver. ApoE is going to bind to this receptor, and this is the ApoE receptor on the liver. And this is going to result in chylomicron remnant being taken up by the liver. Okay, so this is basically the whole life cycle of a chylomicron, so to speak. But there's one more thing that we need to talk about. So this triglyceride, I mentioned that the goal of this chylomicron is to give these triglycerides to the peripheral tissue, right? But in the form of triglyceride, it can't be taken up by the tissue. The same problem that in your GI tract, the triglycerides need to be broken down a little bit first before it can be absorbed. So the same idea happens here. So the triglycerides needs to be broken down into three fatty acids and a glycerol. So in this case, the three fatty acids can now be taken up by the peripheral tissue. And to catalyze this reaction, there's a protein called Lipoprotein Lipase. And that's located on this endothelial wall. So this is basically blood vessel wall. So the pink here is the blood vessel wall. And this protein is sort of anchored onto the blood vessel, and this is called a lipoprotein lipase, abbreviated as LPL. But this lipoprotein lipase isn't active by itself. It needs something to activate it. So remember this ApoC2 here that's given to the chylomicrons by the HDLs. This is going to be activating it's an arrow. So ApoC2 is going to be activating the LPLs, allow it to catalyze this reaction to turn the triglycerides into the three fatty acids, and the three fatty acids is taken up by the peripheral tissue. So green here is peripheral tissue. Okay, so just to recap this whole pathway, chylomicrons came from the GI. First, they are nascent chylomicrons, and they only have an ApoB48 and lots of triglycerides. And then it's going to require a few apoproteins from the HDL, so C2 and E. And after it's acquired those apoproteins, now it becomes a mature chylomicron. The mature chylomicron drops off the triglycerides and drops off the triglycerides to the periphery, and then the chylomicron itself becomes a chylomicron remnant, and the remnant is taken up by the liver. Okay, so I'm going to do the next video.